In multicellular organisms, there are distinct tissues that are often distinguishable by characteristic transcription factors. By identifying promoters under the control of these transcription factors, you can restrict a gene's expression to specific tissues. Additionally, some promoters are only transiently expressed during a particular phase of development, allowing temporal restriction of gene expression. In this study, Liu and coworkers employ a transcriptional control element specific to flower development to control a toxic protein's production. Thus, they can programmatically eliminate flower formation. Homeotic transcription factors are expressed in specific tissue types. Agamus is one of these transcription factors involved in floral organ development. These plant homeotic transcription factors are functionally similar to Hox genes, which define tissue identity in animals, but they are not homologous. Thus, these are examples of convergent evolution. Agamus is involved in formation of stamens and carpels in the third and fourth whorl of the wild-type flower. During plant flower development, there are layers of tissue within the bud, and each radial layer is termed a whorl. In a flower like a rose, the number of whorls is uncontrolled, but in most flowers there are defined numbers of these whorls. In Arabidopsis, these whorls differentiate into distinct portions of the flower, such as stamens, petals, and sepals. AG mutants of the Agamus gene produce flowers with an endless number of flower whorls, repeating the basic pattern sepal, petal, petal. As shown in the figure, there are many other flower phenotypes that can result from mutation of homeotic transcription factors. The Agamus RNA begins to accumulate in flower cells that will later give rise to stamens and carpels. Later in flower development, AG RNA continues to develop in stamens and carpels but becomes restricted to several specific cell types. Thus, Agamus is primarily a transiently expressed product whose most prominent role is during early phases of flower development. The authors put Barnase, which is a potent bacterially derived RNase, under the control of the Agamus gene's regulation to get cell type specific expression of Barnase in flower sex organs. This is proposed as a strategy for preventing the dissemination of transgenes in the environment by creating sterility. The gene structure they constructed is somewhat unusual. On the ends there are targeting sites for a transposase. This is used to transfer the cassette into the plant genome. There is a normal-looking antibiotic resistance marker, and then the Barnase cassette. What is unusual is that the regulatory element being taken from the Agamus gene is the second intron, not some 5' upstream promoter sequence. That intron is placed upstream of a virally derived promoter, then the Barnase and the terminator. The unusual gene structure is due to the means by which the transcriptional control element was defined. It is part of an enhancer trap experiment. The promoter they employ, the CAMV35S promoter, is a minimal promoter that alone is not sufficient for transcription. It has binding sites for an activator, but without the addition of enhancer element, little transcription results. Because enhancers act over long distances through looping the DNA and binding to other sites of mediator, these elements can often be placed at arbitrary positions near a gene and still have the desired effect. In the enhancer trap experiment, the CAMV promoter is placed upstream of a selectable marker or reporter gene. In the illustration, that reporter gene is LAC-Z, allowing blue-white screens. A library of genetic elements, usually from fragmentation of a larger gene cassette, is cloned upstream of the reporter construct, and then variants are screened for tissue-specific phenotypes. From such a screen, it was determined that the second intron contained the enhancer elements that define its tissue localization. Though it is in an intron, this is a transcriptional initiation control element, not some form of splicing or termination control. They introduce their DNA into cells via transposition, and as a result, it inserts into distinct loci. Different integration sites give different behavior. By screening multiple plants, they find some strains that give the desired phenotype, the production of aberrant flowers. In these mutants, the morphology of the entire plant is normal. The flowers have normal petals and stamens, but the stamens and carpels are damaged or absent as predicted. The morphology of the fruits is also aberrant, but they still get made. The plant still produces pollen, but it's morphologically and functionally damaged. The plants also produce seeds that are viable, but their viability in the production of new plants is about one order of magnitude reduced. Thus, it is possible to control a toxic gene and have it ablate a particular cell type, but there are often unintended consequences due to the fact that everything is truly an analog signal, not a digital signal, and rarely are these tissue-specific promoters exactly restricted to one particular tissue type.